Alright everybody, hello. Welcome back to the lecture video series. Today taking a look at lecture 13, which is talking about the first part of translation, so translation 1 in this case. There's going to be a second video on translation after they upload that, but let's go ahead and get into this guy. Lecture 12, recap, did I say lecture? Whatever. We're in lecture 13. Lecture 12 was the reverse transcript day stuff. If you want to take a look at that, go back to the previous lecture. But for this one, we're going to be talking about translation, which is this part here. I'm actually going to go back to using red, because I think red is a bit easier to stand out here. So, go back to that. So, the first thing I want to talk about is ribosomal subunits and what they're made of. So, the ribosomal subunits come in the small and the large. So, there's the large one and the small one. Oh. And the main thing that you should know is that the large one holds the tRNA, and it has the active site for where the peptide bonds form. And it's also the place where the peptide exits as it's being made. So, if the tRNAs come from here or something, and the peptide chain exits from up here or something like that. The cleft is basically where the mRNA and tRNAs are matched together, and the small ribosomal subunit specifically holds the mRNA and the decoding center. So basically where the decoding center is where we match the tRNA to the codon of the mRNA. And if I was to say that's something you should just try and not memorize, but understand that. So understand that the large ribosomal one holds the tRNA. Understand that it's where the peptide bonds are formed. Understand that it's also where the new peptides being formed. The cleft, just understand that that's where the mRNA and tRNA kind of match to each other. And the small ribosomal subunit holds the mRNA, and it's where it's where we try and match the tRNA to the correct codon. From this side, you need to memorize all of these. So please memorize all these numbers and all that stuff. I think it's helpful to know. Use a flashcard, whatever you want. The way I personally remember this is it's 30, 40, 50, 60, 70, 80, and I just know that I personally know that the prokaryotes, the bacterial ones, are always smaller in size than the eukaryote ones, so I just remember it like that. So I say 30, 40, so 30, 40, 30 is bacterial, 40 is eukaryotic, 50, 60, 50 is bacterial, 60 is eukaryotic, 70, 80. 70 is bacterial, and 80 is eukaryotic. That's how I remember it. And S is Svedberg's units. It's a measure of sedimentation time. So basically it's a measure of surface area if you want to think about it like that. So I wouldn't worry about what it means too much though. And the other thing you should memorize is also the rRNA. So how many rRNAs, how many proteins, how many rRNAs, how many proteins, how many rRNAs, how many proteins, that kind of stuff. Or put on your note card, either one. So rRNAs, they have highly conserved 3D folding and modifications. rRNAs are very long. They make up the majority of your RNA in your cells. So there's just a lot of stuff going on. So divergent sequences in yellow, or red. So these sequences are not very conserved, but the green parts are. So most of it's very conserved. And also the folding pattern is very conserved. See how it's pretty much the same across all of these organisms, right? Pretty much. They're also very intensively modified, and it's all just the modifications are important because it helps with the efficiency and accuracy of amino acid and protein synthesis. So just an example of the different types of rRNAs, and there's a bunch of them. So most of them are very conserved. But yeah. Now, hopefully a lot of this stuff is just stuff that you remember from Bio 200, but basically what we're going to do is talk about the large and small subunits and how they kind of like work. So we've got, you know, our proteins, our RNAs. Oh, we talked about this a bit before. We have the mRNA, the tRNA sites, right? We have the A, the P, and the C site. EC site. E. I can't remember the exact names, but it's like it's just written here. Yeah, so amino acyl site, peptidyl transfer site, and an exit site. So basically what's going to happen is a tRNA, right? So as you read the mRNA within the codons, right? Each amino acid, right, is going to be brought in by a tRNA. It's going to go to the A site, the amino acyl site. And then it's going to go from that, it's going to switch to the P site, which is the peptidyl transfer site. So what happens there is that the amino acid is attached to the growing peptide chain through a transesterification reaction. And then the tRNA that has lost its amino acid goes to the E site, and it is kicked out of the ribosome. So remember that to make each one of these amino acids, right, these amino acyl tRNAs, you need to use 1 ATP. There's also GTP actually that is used, and we'll talk about this a bit more later. But the GTP is used to drive the translation check and to make sure that the correct amino acid is installed when it's supposed to be. Here's just another image of what it looks like. Don't think it's very important. Just remember we've got the A site, the amino acyl tRNA site, the P site, the peptidyl tRNA site, and the exit site. This needs to be memorized. All of it. So, first remember the activation of the amino acid, right? That's the amino acyl that's the tRNA synthetases, right? So this uses ATP, magnesium cofactor, because ATP, right? If you have ATP as a thing, you're probably going to have magnesium as a cofactor. For initiation, this is in your ribosome specifically. You need your mRNA. You need a FMET tRNA, which is methionine, right? So methionine is the amino acid that initiates translation at the start codon. And you also need your small ribosomal subunit, 30S in the case of prokaryotes, 40S in the case of eukaryotes. You also need your 50S or 60S, and all 3 initiation factors plus a bunch of GTP. Right. For elongation, similarly, right, we need the full complex, 70S or 80S in the case of eukaryotes. You need all of your amino acyl tRNAs and their specific codons. You need these 3 elongation factors, EFTU, EFTS, and EFG. And you need more GTP and magnesium. And then for termination, you need your 3 release factors, or 4 release factors. There's release factors 1, 2, and 3, and then there's ribosomal release factor. And you also need EFG again and IF3. So these all need to be memorized, right? And the way I would memorize these, actually, you can use this table if you want, but there's a more detailed description or more detailed images of what happens down below, so I'll go over those. And then after translation, you have like chaperones and folding and stuff like that. So, yeah. I'm not going to go over this. You have an animation here if you want to look at it. Feel free. This is also a little bit complicated. You can follow this if you want, but I'll just go over them. Like, this is the whole process. But let's go over initiation first. So, the first thing that we need is we need our N-FMET tRNA, which is our N-formal methionine tRNA in bacteria, and our methionine tRNA, just normal MET, in eukaryotes. They usually don't ask specific questions about the type of MET required, or methionine required, but the only difference between bacteria and eukaryotes is the addition of this acetyl group, basically. So, yeah. The other thing that you should keep in mind is this difference. AU in eukaryotes and CA in bacteria. But remember, this always has that CCA motif, right, from what we talked about in the previous video. So there's CCA motif up here. But yeah. There's this enzyme here, transformylase. For bacteria, it transfers the formal group, this acetyl group, from tetrahydrofolate, from THF onto the tRNA here. So, we've got, so initiation first assembles a pre-initiation complex. So, in prokaryotes and eukaryotes, we have slightly different ways of how this works. But the way you should try and, like, I guess, like, remember if you want to think about it like this is, like, first we have, like, the mRNA shape. And in bacteria, right, it's just linear. But in eukaryotes, it's actually U-shaped. That's because we involve a bunch of other proteins, like these 4E and these polyadenylate binding proteins. Because we have a poly-A tail, right, in bacteria. We don't have a poly-A tail in prokaryotes, right? So, eukaryotes have this kind of U-shaped mRNA structure. And then for the mRNA sequence, right, we just have our 5', there's no cap, the Shine-Delgado sequence, and then the 3' end. I'm not gonna lie, I cannot remember off the top of my head how the Shine-Delgado sequence is important here. I believe, if I'm not mistaken, it's basically the ribosomal binding site. So this right here, this RBS, that's the SD. This, I think it's Shine-Delgado, not Shine-Delgado. But the SD, the Shine-Delgado sequence, is the RBS, so the ribosomal binding site for prokaryotes. In eukaryotes, we don't have that. In eukaryotes, we have MCGPP, which is the 7-methylguanosine cap, right? The Kozak sequence, which is like the ribosomal binding site in eukaryotes, and then we have our poly-A tail. So that's what this all stands for. So now we get to talk about the different, the GTPase. The GTPase just means a thing that uses GTP and hydrolyzes it. That's all it means. So, first we have our initiator tRNA, which is our methyl tRNA. Then we have our mRNA, or AUG, and our other codons. We've got our small ribosome plus initiation factors, which is what IF stands for. We have our IF GTPase. And the GTPases, they serve as like the switch that triggers translation, if you will. So basically what we're going to take a look at here, I'm trying to see if I can find the specific guys, but does he explain which ones are which? I believe, if I'm not mistaken, IF3. Let's see. So, if I'm not mistaken, so IF1, 2, and 3. So I can't remember which ones are which exactly. But I was trying to see if he maybe showed you guys which ones are which, but doesn't look like it. Basically these, right, these, this is one of them, so these are all three of these are the initiation factors you need to know. So it's IF1, IF2, and IF3. I don't know which ones are which. He doesn't tell you which is. I believe, if I'm not mistaken, this is IF2. IF2 is the one that acts as the GTPase. I'm gonna try and see if I can Google that real quick. But there was like a bro. Let's see here. Yeah, okay, here we go. So, here, this is why I hate these lectures sometimes. They don't, like, talk about which ones are which. This is IF1. So, IF1 Can I increase the size of this? IF1, IF3, IF2. And basically what you need to know is that IF2 cannot bind unless IF1 is already bound. So IF2 and 1 attach to each other, and IF3 kind of helps lodge the mRNA on top of the small ribosomal subunit. So basically what's happening, right, is we have this happening here. The GTPase, right, serves as a switch to trigger it. When IF3 will bind with this mRNA to kind of anchor this, right? And then when that happens, right, the IF1 will bind, and then IF2 will bind on top of IF1. And then that will allow the tRNA to kind of add on to this. And then once all of these pieces are in place like that, this GTP will get hydrolyzed, and that hydrolyzation will cause the large subunit to slap on top. And then all of these factors will dissociate. So if I was to remember one thing mainly from this, it would be that IF2 is the GTPase, but you need to remember this entire process, basically. So you should remember that, like, oh, you know, to start this process, I need this methynyl tRNA, the mRNA, the small ribosome plus the factors, and IF2, the GTPase. And this takes one GTP, right, to initiate this whole process. So, again, just remember that, like, the way this happens is, right, I have my small ribosomal subunit, the mRNA attaches on, the IF3 secures it, the IF1 then attaches, and this IF1 allows IF2 to attach on to IF1, and then when IF2 hydrolyzes GTP, that causes the large ribosomal subunit to attach on top. Then we move on to the elongation phase, right? The elongation phase is a little bit different. But in this phase, what's happening is that we have EFTU, which is another GTPase, and this EFTU is what's responsible for bringing the different tRNAs. So there's two options here. If EFTU brings an amino acid that has a tRNA anticodon that is complementary to the mRNA, then it's a cognate or a near cognate. If it's a non-cognate, right, it doesn't match at all. It goes in the other direction. But let's talk about what happens if it's a near cognate, right? So if it's a near cognate, or if it's a near, it's close to a cognate, or it is just a cognate. So cognate means it exactly matches. A near cognate means that maybe the last base pair is missing, right? Or maybe the last base pair is not the same. So, right, so if it's a cognate, we're going to take our EF2 GTP, and we're going to go to the next slide. And what's going to happen here is that once this thing attaches on, EFTU is going to hydrolyze its GTP, and that's going to be the way that the ribosome basically checks to see if the amino acid is correct. It's basically like a clock. So if by the time the GTP is fully hydrolyzed, the tRNA is not properly attached, then the tRNA will just fall off and leave, right? And the amino acid will never be attached on. But if by the time the GTP is hydrolyzed, the tRNA and amino acid are like properly locked on to this complementary codon here, then the EFTU just leaves with a hydrolyzed GTP, and because the tRNA is so strongly bonded to the codon, it can't pull the codon. Sorry, it can't pull the tRNA off with it. So the EFTU just leaves on its own with the GDP. So what's essentially happening is, right, we start with our ribosome. We have our EFTU with our GTP. If the amino acid is a cognate and the codon is the anticodon is a cognate, right, we move to the next phase. And now the EFTU tRNA complex, right, with the GTP attaches on to the codon. Then the GTP starts to hydrolyze. This GTP starts to hydrolyze. If by the time that GTP is hydrolyzed, the tRNA is not properly bound into this section of the mRNA, due to that loose binding, this tRNA will just leave alongside the EFTU, right? If it does properly bind after the GTP is hydrolyzed, then just the EFTU is what ends up leaving. So if we go here, right, see here in this image here, the GDP is hydrolyzed, right, properly hydrolyzed. If it was a cognate, then this would be properly bound to the codon, and so the GDP is just going to leave with the EFTU. And then everything's going to shift, right? Everything's going to slant and shift over, and then you're going to start this whole process all over again. If it's a near cognate, right, then usually what ends up happening is the near cognate probably doesn't bind super strongly, and so if it's not binding super strongly, when the GDP EFTU dissociates, the tRNA isn't bound strongly enough to the mRNA to keep it stuck there, and so it just floats away. Right. Back here for the non-cognates, right? If you try and bind the non-cognate here, it just won't even fit in the first place. So it just goes back the other way. Right? If it's a non-cognate, it's not the correct sequence. It's not even going to bind at all, pretty much. So it's going to try and bind. It's going to try and go this way, right? But then it's not going to match, so it's just going to go back the other direction. So what you should remember from this, right, is that each amino acid that you try and add, right, uses a GTP. So even if you get to this point and it's not the correct amino acid being added, it still uses up a GTP for the fidelity check. So and you can see here cognate is fully accurate, near cognate 1 mismatch, and non-cognate is more than 1 mismatch. And the steps are exactly as listed here. So the first thing that happens is the rapid reversible binding of EFTU aminoacyl tRNAs, right? So these bind to the site. Then the stabilization of the cognate aminoacyl tRNAs and the aminoacyl tRNAs. Then the GTP hydrolysis occurs, right? The hydrolysis occurs here to see if it actually is the correct amino acid. And then the dissociation of EFTU. And then if it's the correct amino acid, the tRNA stays stuck. If it's not the correct amino acid, it just floats away. In this case here. Here is just a nice little image of what it looks like in terms of the mRNA, tRNA, and stuff like that, but I'm not going to bother with that. Here's just like some stuff about how the peptide bonds are formed and all that stuff, like the catalysis of that. But yeah, basically, because the amino acids are already activated by the aminoacyl tRNA synthetases, you don't need any extra energy, right, for that transfer step. So for the tRNA, literally all you have to do is you just got to basically you just use the tRNA of one amino acid, right, and you use it to attack the next one, right? So you can see up here, there's one tRNA here, another tRNA here. The amino acid, the N terminus of this amino acid attacks the C terminus of this one, right? And this leaves, right? Okay, now for the translocation. So translocation is like, once we've attached our amino acid, we have to shift everything over so we can repeat the process again. So it basically what happens is like everything moves, right? And then like this, and then shifts down, right? So I wish I could show this in person, but I don't have a way of showing this. But it's like, it shifts this way, and then it pulls the bottom half along with it. And so that allows for a new tRNA to come into this spot here. And so the factor we end up using for that is EFG. And EFG also uses GTP. So every time you do a shift like that, you use another GTP. So EFG and EFTU are actually very similar, right? So EFTU tRNA is like a thing that uses GTP, right? Remember, this is the one that does this fidelity check up here. This guy, ooh. This guy right here, this is EFT, GTP, amino acyl tRNA, right? So basically, right, these two have similar mechanisms. Both are GTPs, but for the translocation step, where we're shifting everything over so that we can add another tRNA, right? So we can add another tRNA into this spot, right, to continue the process. That shift requires GTP for EFG. That's it. That's all you really care about for this slide. That process is called translocation. And now we're on to termination. So, termination recruits release factors. Release factors are proteins. They kind of seem like tRNAs, but they're not tRNAs. They bind to stop codons. So basically what happens is, right, we've got class 1 release factors. They're proteins. They mimic tRNAs, and they bind to the A site like tRNAs do. And what happens is residues in the anticodon loop of the tRNA, or not tRNA, of the release factor, are able to recognize the stop codon. And so what happens is you have this GGQ motif, which I believe is glycine-glutamine, if I'm not mistaken. This GGQ motif is really important because actually what it ends up doing is it allows for the coordination of a water molecule in the P site, right? So the P site is the peptidotransferase site. Normally what happens in that site is that one amino acid, right, will do a nucleophilic attack on the next amino acid so you can form the amide bond, right? But when you use the release factor doesn't carry an amino acid. Instead, it carries this GGQ motif, which is kind of, it's interacting with a water molecule. And so instead of the N-terminus of one amino acid attacking the C-terminus of the other, the water molecule ends up attacking the C-terminus, and you end up with the carboxy end after this termination has finished. And here's some structures. I don't think this is that important, but these are all RF1s, release factor 1s. And here's the anticodon loop as shown here. This is the part that binds to the stop codons. So, termination recycling. So basically what's going to happen, right, is we've got RF1 and RF2, right, RF1 / RF2, either one. When the stop codon is there, right, the release factor will bind to the stop codon. And then that binding causes the association of that water molecule with the GGQ motif to the P site, right? And that basically leads to the cleavage of the peptide. So the peptide floats away, right? So once the peptide is gone, right, we then use this RF3, which is another another GTPase, right? And this GTPase, what it does is it attaches on, right? And that attachment, and then hydrolysis, right, of this GTP causes the release factor 1, and everything to fall off. So once it's hydrolyzed, once it's hydrolyzed, right, the release factor 1/2 and release factor 3 both kind of fall off, and then you can use the ribosome again, maybe for another cycle of something else, right? Maybe a different mRNA has to come in or whatever, right? So that's termination, right? So once you've done that, everything is done. If you, however, want to recycle, when you start doing the recycling, right, the recycling process basically starts from this point here. So we take this point, right, and we move to recycling. So now, in the recycling stage, what we have to do before we're able to actually reuse this ribosome is we have to split the large ribosomal subunit and the small cell ribosomal subunit. And the way that we do that is through RRF, the ribosomal release factor, and the EFG. And I don't like how he's done this, where he's like put this on top of whatever. But basically, you can think about this as a crowbar, right? So our ribosomal release factor, it basically binds to the A site along with EFG. And what you're basically doing is using GTP the hydrolysis of GTP on EFG, which EFG, by the way, is the same guy that you used up here. Using the hydrolysis of EFG, what you do is you basically push on EFG, so the hydrolysis of GTP allows EFG to do work, and so it pushes down, which causes RRF to move up, if you will, and then that movement of that upwards causes the large ribosomal subunit to kind of disconnect from the small ribosomal subunit. And once that's done, right, everything gets released, the small ribosomal subunit and the large ribosomal subunit split. So there's two GTPases in this case, right? So release factor 3, right, uses release factor 3 is used in the first part of termination to so release factor 3 is used first, right? So after RF1/2 binds, right, and causes the peptide cleavage. RF3 binds, right, and the hydrolysis of RF3 causes RF1 to leave. RF1/2. And then we go into recycling, right? For recycling, what happens is we use EFG and RRF. EFG acts like a crowbar using GTP, and then it basically pushes down, which causes RRF to move upwards, which splits the ribosomal subunits apart. And then everything leaves. Kinetic proofreading, we already talked about this, right? Slow to hydrolyze variant of GTP. Basically, this idea of slower GTP hydrolysis results in higher accuracy. Hopefully this kind of makes sense, because like if you slow down the amount of time it takes for the hydrolysis to occur, there's more time to double check that the tRNA actually properly binds to the codon, right? So there's more time to check if those bonds are actually forming, right? So if you give it more time to check, then it should be more accurate, right? So that's kind of the main idea there. I hope that makes sense, right? What they're saying is that they did an experiment where they made this GTP variant that's slower to hydrolyze. And what they basically found is that, you know, when you slow down the hydrolysis of GTP in the EFTU step here, that allows the ribosome to check if the tRNA matches properly more, right, for a longer period of time. And if it, you know, if you give it more time to check if it accurately matches, right, there's going to be more chances for it to say, oh, wait, this matches perfectly, or, oh, wait a minute, this actually doesn't match very well, right? So it gives it more chances to kind of kick off any tRNAs that don't properly fit. Okay, and then for quality control by early termination, this is just, you know, a way of saying, if too many non-cognates bind and they somehow get stuck in there, it terminates early. That's it, right? So, like, for example, if a low fidelity, so maybe the A site, right? If you bind a tRNA, right, and the tRNA doesn't properly match the codon, right? Usually what happens is it gets kicked out, right? But maybe there's a case where it doesn't get kicked out. And so then you bind the next tRNA, and that also doesn't fit properly. And so under that situation, the ribosome will recognize that, oh, wait a minute, I'm just binding a bunch of stuff that doesn't work. Let me just dissociate myself so I don't make a bad protein. And so what ends up happening is the release factor 1 and the ribosomal release factors, these guys from up here, ribosomal release factor 3, so RF1/2 and then RF3, they bind, they hydrolyze, and then they dissociate the ribosome, basically. So this happens a lot under stress conditions, for example, like starvation. This makes sense, right? Because if you have starvation, right, it can lead to loss of certain tRNAs. So if you're missing tRNAs, this happens a lot. And so if you're missing tRNAs, right, it makes sense that the ribosome would try and basically kill itself to stop itself from producing faulty proteins that could potentially harm the cell. So, yeah. But yeah, that's actually the end of this video. I hope it was helpful. Kind of went through this gun a bit quickly, but I guess it's 34 minutes. It's not that quick, but I hope this video was helpful for you guys. I'm going to have a workshop on Tuesday, I think. Hopefully I'm feeling a little bit better by then. A lot of content to cover for these exams. This one's a bit of a... Exam 2 is a bit of a tough one, but yeah. Hopefully these videos are helpful, and I'll see you guys in the next one. See ya.